Hello, and a very warm welcome to everyone joining us for today's Select Science webinar, using direct CIEF-MS and Prep-CIEF-MS for MS-characterization of protein isoforms, LC-MS analysis, peptide mapping, and other techniques. My name is Sarah Thomas, and I will be moderating today's presentation. I'm delighted to be joined today by three presenters: Professor Christian Nozus, Dean of Analytical and Bioanalytical Chemistry at Ulm University; Carsten Sunksen, a Senior Researcher at Novo Nordisk; and Tiemen Huang, President and CEO of Advanced Electrophoresis Solutions. Today, our speakers will discuss the application of CIEF-MS for protein isoform characterization, the use of Prep-CIEF with subsequent peptide mapping and LC-MS analysis, and we'll also learn about the experimental setups needed to run such experiments. After the presentation, we'll move on to our question and answer session, so please feel free to ask any questions for the Q&A session at any time during the webinar, and you can submit your question to the left of your screen. So without further delay, I would like to hand over to our speakers for today's presentation, and I would like to thank them all for presenting to us today. Please go ahead, Tiemen.
Thank you very much, Sarah, for the introduction. Thanks Martin of Isoform Life Science to organize the webinar. Hello, everyone. I'm Tiemen Huang. I will try to brief you some exciting new instrument setup and application of whole column image detection capillary isoelectrical focusing, or CIEF, in about 10 minutes. Advanced Electrophoresis Solutions is located in Canada Technologies Triangle, Waterloo region. So we established 10 years ago, and we have been working to provide a total solution for high efficient CIEF protein separation and fractionation. The system we made is based on whole column image detection. Essentially, a 5 centimeter long capillary is monitored by an image camera, and the whole separation process can be monitored in real time. The CIEF have a good application for protein isomer separation. However, until we develop the fractionation cartridge, there's a great challenge to isolate the isomer peak and do the MS coupling. This figure will illustrate the schematics of how the fractionation or MS coupling work with our system. We actually manufacture the way with a very large inner diameter separation column and a very small inner diameter transfer capillary. So once the protein focused, a syringe pump is pushed to focus the protein toward the right end of the transfer capillary. Because of the unique proprietary column inner diameter transformation technology, the focused peak once inside the transfer capillary will be stretched out. This eliminates the potential peak mixing with the other method. There's other advantage associated with the larger inner diameter separation capillary. For example, the higher detection sensitivity and the higher sample load. I'll show the focusing and the mobilization process with a movie. You will see the protein with 2 pi marker was focused in the system. So it's not like what you see in the other a chromatograph. Is the intensity at a different time. Essentially, this is the intensity along the separation column at a specific time. So you can see the peak, the protein focused and mobilized toward the right end of the separation column. So you will see the profile of the protein is essentially the same as the focus. For preparative CIEF, the transfer capillary is connected to the inlet of a 10-port selection valve. So once the focused peak pushes outside the transfer capillary, it will be collected sequentially by the different vials. This figure shows analytical CIEF of a mat. So based on the same method, we actually use a larger inner diameter, 320, to fractionate the isomer peaks. And the peak was collected into individual vials and mixed with the isoelectrofocusing matrix and re-inject into the analytical CIEF for peak purity confirmation. You can see the basic peak, the mean peak, and assay peak quite like pure peak was confirmed. I won't go into much detail on the usage of these peaks, and this will be introduced by Dr. Sungsen later on. For MS coupling, you can do several ways. Either as Professor Newsys will be introduced later, the transfer capillary can be inserted to a nano sprayer and do the nano ESI MS. Or you can introduce in like the make-up liquid just after the peak migrate out of the transfer capillary, and the eluent will introduce directly to the ESI source of any mass spectrometer. This figure shows 6 times separation of the NIST map. We can see the good separation and the reproducibility were obtained with the CIEF. Still, based on the established method, we can seamlessly transfer the method to CIEF-MS. With like this experiment is with a 200 micro MS cartridge. So we can see the resolution with the 200 is about the same with the even like lower amperlite concentration. And we can see once the peak migrates into the MS, you can see the order is reversed. The basic peak come out first, but the comparable charge variant pattern was obtained for the UV detection and the CIEF-MS. The signal for the MS is very good, so all charge variants of NIST map were deconvoluted, and the different isoglycal form of NIST map can be identified, and also the mean peak and the 2 basic peaks were identified based on the norm mass and the mass difference of multiplication. You can notice that the acidic fractions, the MS is quite complicated, and this will be done for fractionation and for ambiguous identification later on. So AES is provided the other like the coating for the separation, such as the hydrophilic one for the minimize the use of methyl cellulose, and also we provide a narrow range carrier amperlite, for example, like a 1 pH unit to increase the separation, so that when we do the preparative CIEF or CIEF-MS, so the good resolution can be obtained. So there's many advantages associated with the CIEF, preparative CIEF, and CIEF-MS. I'm confident you will add more on the list if you be using this technology. In essence, the CIEF-MS is the modern form of 2D electrophoresis. There's many advantages associated with CIEF-MS, and we would like to work with you to explore the application in your proteomic studies. This picture shows the configuration of our system. The CE instrument is to the right, and the autosampler, the syringe pump is on the left of the system. So essentially, you can use the system to do the analytical CIEF, you do the isomer charge peak collection, and also do CIEF-MS. So with this, I conclude my brief talks. Thank you very much for your time.
Okay, thank you very much for your kind introduction. And this is Christian Neuses from our university in Germany. And I'm going to talk about our recent results on coupling the CE-infinite system to electrospray mass spectrometry. And the main person involved in this is Johannes Schlecht, a PhD student in my lab. And before starting with the real subject, I want to give a very short motivation on that, why we are doing it. And probably you're familiar with the heterogeneity, what can come out from processing and production of and impurities arising during storage from monoclonal antibody. And what you see here is a lot of possible variants, and most of them change charge. And this is why we try to have charge-based separations, as you have seen in the previous presentation already a little bit. You might think about, well, mass spectrometry can do a lot nowadays, and that's right. But if you think about these huge molecules like trastuzumab, that's really difficult to separate by other systems, and mass spectrometry still is difficult on these huge molecules. But you will see that you can do a lot if you have done some separation, as with the isoelectric focusing. In many cases, to separate charge variants or other methods used in electrophoretic or used as electrophoretic techniques, you have a lot of molecules involved, like in CZE, different buffer components in isoelectric focusing, like coupled analytes, ampholites, etc. And in CESDS, of course, many more, which will interfere with electrospray ionization mass spectrometry. So how to couple these systems still to MS is not that easy. And that's what I will explain how we did it. Indeed, all these techniques are widely used, and that's, of course, a key point. And about 3/4 of all FDA-approved biopharmaceuticals in the last years have used such CE-based methods, but hardly any of them has been used in coupling with mass spectrometry. And definitely they need to know what is in, because that's, of course, a key point regarding identification. And from these electrophoretic-based methods, about roughly half is about CESDS, and 1/3 is nowadays, and this is rising a lot, is capillary isoelectric focusing or imaging capillary isoelectric focusing. So how can you couple this to mass spectrometry? We have been developing since many years now, or some years now, I would say, a two-dimensional system. So having in the first dimension whatever CE system you might use, including capillary isoelectric focusing, and then cutting out a peak of interest with a small internal loop in this nanoliter valve and transferring to the second dimension. And this looks like this at the moment. So you have coupled two CE instrument in front of the mass spectrometer, and in between we have the valve. To look in more detail, you will see on the right-hand picture the valve we have been using. So basically just 4 connections and an internal loop. And the loop itself is seen on the next picture. It's the small, U-shaped, tiny little structure in there, which is about 20 nanoliters. In most cases, we use it in this size. All right, so how did we do that? How did we apply that? And just very recently, a couple of weeks ago, a paper appeared from our group, which showing that you can even couple CESDS in this two-dimensional setup, as you have been seeing. But that's not the subject today. You see just that this is possible as well, and this giving you the size heterogeneity of impurities of antibodies in a very detailed way. Now I will come what you can do in doing the same with isoelectric focusing, and in particular using imaging capillary isoelectric focusing. And the first approach we did is coupling the imaging capillary isoelectric focusing, as you see in the upper picture, directly to our QTOF mass spectrometer, we have been just using the valve for cutting out the peak of interest and then using a syringe pump to flush it over to the mass spec. So it's some kind of flow injection in the second dimension, not really a two-dimensional setup. But we use the two-dimensional setup as well. In this case, on the right-hand side, there's again the CE infinite. On the left-hand side, we have an additional dimension, and now we use our CE sprayer, of course, because we are applying high voltage in our second dimension going via the valve to the sprayer. We use this both for the same molecule, and you will see results from that. So in the next slide, in the middle, you see the profile, what you get from trastuzumab with a small basic variant, the main compound, of course, and then acidic variants, as you have seen in the previous presentation already. Now cutting out the main peak, using on one hand side the direct infusion valve, you see the spectrum on the left-hand side. That's the antibody charge distribution you obtain. And if you do it in the two-dimensional setup, you see this much cleaner. The spectrum is much cleaner than with a flow injection system, so you don't have these tiny little peaks on the left-hand side. And indeed, the mass spectrum, the deconvoluted one, is much purer. If you compare this, on the right-hand side, you see the deconvolution from the two-dimensional, on the left-hand side from the direct infusion, and you see still some adduct formation in the direct approach. This was using hydroxymethylcellulose at that time, and later on we could avoid this because that's what is causing the interference here, which we separate out if we have a real two-dimensional system. Of course, not the main peak is of interest. We are interested mostly in the variants, and have a look on this small tiny peak of the basic variant, and indeed it was possible to characterize this pretty well. So if you cut the basic variants there, you will see the deconvoluted spectrum on the right-hand side, and the quality is good enough to see the glycosylation pattern. And indeed, we have a shift in mass of about 18, 19 Dalton, which is corresponding to succinimide formation or pyroglutamate formation or whatever, modifying the protein that it's getting, one amino acid that it's getting a basic form, a basic variant here. All right, with this in mind, we of course thought, how can we make it simpler? And the next idea was, can't we do it directly without any valve, without a second dimension for purification? So the idea was to simplify our setup by directly using to transfer capillary to electrospray MS. However, from the solution used in isoelectric focusing, you have a water-based system with ampholytes and, of course, your analyte in there, which is not very ideal for spraying. So what we need is definitely to add an organic modifier to get proper ionization efficiency. So the question is how to add it, and there are two possibilities. And the first one is to add it right in between our transfer capillary. The question is, can we maintain the separation of our imaging capillary isoelectric focusing system in a way that we get for each peak the result of in the mass spectrometer afterwards. So we took again trastuzumab, as you have seen, and we used a standard system set up optimized for trastuzumab, like a Pharmalit mixture, in this case 2 milligram per milliliter trastuzumab sample, and with this we obtain again the separation as you have seen before, and with this the mass spec or the take of the mass spectrometer was not very successful, but still we could get a reasonable good mass spectra out there where you can identify all the glycoforms on the main variants. However, it was not possible to characterize the basic and acidic variant in this setup, so we lost our separation. So we went on to a second possibility. Rather than coupling the organic in between, we used an CE sprayer, a nano-sheath liquid interface, where the sheath liquid is grounding or contacting our capillary end. And in this way, we ionize right at the end and do not introduce anything in front. And we used this setup in combination with our Orbitrap Fusion Lumos. And you see here on the right-hand side a picture, a photograph of the transfer capillary inserted into the emitter. All right, how does it look like? The result, what we get is that is the profile of the main peak. You see again the parameter, which are the same as shown before, and taking the main peak, you see the profile on the left-hand side. You see the profile of the mass spectrum on the right-hand side, and indeed, it's not perfectly ideal, but you still see the basic variant, which is coming here, of course, leaving first the capillary, coming in first in the MS tick, and then you have the main peak, and then you have acidic variant. So we got mass spectra out of these 4 regions, let's say, and have had a look whether we can characterize our sample in this way. And indeed, the mass spectrum looks very clear, and if you then deconvolute it, you end up for the main peak with a detailed description of your glycoforms. Even the small variants showing on the left-hand side, you see only one glycosylation site. Even those could be detected in very much detail, so you can get exact glycosylation profile here first. But of course, what is with the other compounds? And indeed, if you look in the basic variant, you see a similar profile, but shifted towards higher masses, but we still get all this detailed characterization even on this minor peak. And if you compare the masses and taking all the glycosylation pattern, we end up for the acidic variant with about 1 Dalton plus, and for the later on something like 2 Dalton plus, looks like a singly or doubly deamidated form. And the basic variant is minus around 19 Dalton, which would fit to either succinimide formation or pyroglutamate formation, as we have been discussing before. So this is really like a powerful tool, because you can directly couple your CIEF-MS, your imaging capillary isocyclic focusing system, using this nano interface to mass spectrometry, and can get mass spectra of all of these variants, although the separation is not perfectly retained, so we have to work on that this is getting even better, so that you get the same peak profile in detail as in the mass spec as you have been obtained before in the imaging capillary isocyclic focusing. So to conclude, I think you have seen that our approach in the 2-dimensional setup is working pretty nice in general to characterize impurities or unknown peaks in non-CE compatible methods to mass spectrometry. And this has been showing also for imaging capillary isocyclic focusing. As you have seen, even the minor variants are successfully characterized. But going a step further, if you do a direct approach without a 2-dimensional system, this is possible if you have the right system, which means no additives beyond your ampholytes, and this works, that you get even the characteristic of minor isoforms, though we have to improve the separation a little bit further. In total, I would say this is a powerful tool for biopharma, and I guess Carsten will later on give you some more potential applications. With this, I would like to acknowledge my group, especially Johannes Schlecht, who did the direct coupling work in the last year, about Jennifer Römer, who did the CESDS CE coupling, and Cristina Montalegre, my former postdoc, who did the initial work on the imaging capillary isocyclic focusing coupling, as well as Steffen Kiesig and Bernd Moritz from Roche for continuous and very important support of our technology development. Thank you very much for your kind attention.
So thank you very much for the introduction, Sarah. Well, I will continue in a different way. While Christian showed inline connection to MS mass spectrometry, I will show some initial results from offline analysis of the fractions. This will be by several methods. It will be by intact mass analysis of the heavy chain and the light chain, and half antibodies, as well as peptide map characterization. And this is one of our company's own antibodies, which today is called antibody X. And just to highlight again why we do this, I think Christian already mentioned it. Just add up a little bit. So an analysis and characterization of the charged isoforms is a request from the authorities. This is why we do it, and of course it's also a a need in order to guarantee the patient safety. And the charged isoforms origins of a heterogeneous product, and as well can charged isoform patterns be an indication of degradation. But this is why we want to, as a company, investigate them further in a proper way. And I think Christian already mentioned that acidic species can be generated due to demidation, fragments, glycation, sialic acids, reduced isoform bonds, etc. And basic species can be due to terminal amino acid, like lysine, glucine, isoasp, and some more things as well. Just to start up again, this is how an electrophotogram looks of our reference antibody X. It's a little bit overloaded one. You see it on the main peak in the middle. And then you have the basic one to the right, basic 2, and to the left, acidic 1, acidic 2. And just to remind you of what Timon showed before, here we have the principle of the preparative ISEF, where you have the focus proteins, which then are pressed out by a very low flow. And here I have to mention that, contrary to what you normally do, these samples or these single fractions, they are collected in a volume. In our case, it was in a volume of 10 microliter of a solvent of our own choice. So we took some buffer, some amino acid buffer, and the flow for the elution was about 0.16 microliter per minute. So this means that not much volume is added to this 10 microliter. And this could be repeated several times in order to obtain higher concentration for the single fractions. And like Timon also mentioned, of course, we then checked the purity of the fractions. And here is an overlay of the different fractions. You can see the main form in the middle is very nicely separated. I was very surprised. This is one of our first or initial experiments to see how little carryover there is from the basic 1 and acidic 1. So the main form is almost pure. Basic 1 as well, there's almost minimal carryover from the main form. And even the basic 2 is also very clean. Acidic 1 as well, it's also clean, almost no main form present. And between acidic 1 and acidic 2, there's just a little overlay. So we were very satisfied with these results. And since this has been, the fraction collection has been done manually, these samples were of factors were of high value for us. So we tried to get as much as information out of these samples as possible. And we did it in 2 series. One series was for the intact and subunit mass analysis. And then we performed a second series for the peptide map. So I just want to show some data for the intact and the subunits. So, and here the MS analysis was performed on a Bucamaxis, and it was a 1 millimeter column with 150 millimeters length, C4, formic acid acetonitrile gradient. And here we got data for some of the fractions, again because we had to direct only a little bit of aliquot of this one for the intact. But here you clearly can see that the main, the acidic 1, acidic 2, basic 1 and basic 2, there we got too little signal, but still for the other ones, very nice signal is obtained, and you don't need a nanosystem in order to obtain data of your samples. And I mean, in another way or another trial, we would of course take more of the basic 2 and basic 1 fractions in order to obtain data for this one as well. Another feature I want to highlight is the purity of the sample, because one thing we were concerned about is how do the ampholytes interfere with the mass spec signal of our fractions. And here, just take one example to show that the mass spectra are almost free of noise in this area at least. So it's a quite clean system where good raw data can be obtained. After injection for the intact mass, we added some DTT in order to reduce them, and only limited disulfide reduction was obtained. So this resulted in half antibodies, which are here, which you can see here. So here we have the main peak, acidic 1, and basic 1. And there are some indications at the mass 7440 that this could be a lysine on the basic 1 relative to the main on the acidic 1. This will have to be investigated later on again. And again here, just to check for the purity of the MS raw data, again, very nice peaks are obtained, very clean, with only limited noise from ampholytes. Then here we have some data on the heavy chain. Again, in this case, the signal to noise of the deconvolute data is a little bit limited due to the lower amount of sample we added, but it's still possible, and for sure something we will do further on for this and other projects. And then the next step was to perform detailed peptide map analysis. And we did this with CAP-LC-MS as well as nano-LC-MS. Here I'll only show the CAP-LC-MS because it showed up that the CAP-LC-MS was sensitive enough for the samples we got out. And this was performed with a thermo-easy spray column, 150 micrometer inner diameter and 15 centimeters long. And it was a thermo-CIEF orbit trap mass spectrometer. It's just to show you that data, some nice chromatograms were able to come out of these analysis. So for sure their sensitivity is good enough, and due to the robustness, or the improved robustness on the CAP-LC, we will continue with the CAP-LC system rather than the nano-LC system. Just some results we obtained. So here's an example of the quantification by the CAP-LC-MS peptide map. And this is on the terminal lysine on the heavy chain. And there you can see that the basic 2 has a high degree on lysine present, while basic 1 has less. It's about 12–15%. And the main acidic 1 and acidic 2 forms almost have no lysine present on the heavy chain. So, and here I want to conclude that first of all, we were very happy with the nice and clean separation of the different isoforms within the fractions, even though it was manual sample preparation. And then we were able to show that it's possible to do intact mass analysis as well as heavy chain, light chain, and half antibody analysis, and subsequently as well as peptide map analysis. And this was even possible with a CAP-LC system, which now has shown to be more robust than a nano-LC-MS. So this for sure will be used in several projects in order to characterize the different isoforms we have within the projects. And here I want to thanks a lot of my colleagues, intern and outern. Marika, who is our expert on the ISAF, and Birgit, Marika and Birgit, both working on the system. Martin for introducing us to the system and providing us with help. Jennifer, and then I got some help from Lilia and Jonas on the capacity system. Thank you very much, and thank you for listening to my talk.
Great, thank you for those very informative presentations. It's now time for our question and answer session. So if you have any more questions, please submit them to the left of your screen. So our first question here for Carsten. What does this technique bring uniquely for you?
Hello, can you hear me?
Yeah.
Well, we had several cases where, well, let me start in another place. Image capillary ISIF we use for release test, and we had several cases where it was difficult to analyze the charged isoforms by complementary techniques like ion exchange methods. So it was very hard for us to align or to transpose these results to capillary or image capillary ISIF. So this is why actually fractionating them had a high unique impact on the way we could analyze our samples. And right now we have no other choice than doing this, and I was very happy to see how nice it worked out. Yeah, so great technique, great possibility.
Great, thank you, Carsten. The next question we have here, this is for Christian, I believe. How reproducible is this direct coupling to be made day to day?
Well, the coupling directly is very reproducible, and imaging CIEF is anyway. The only point is that there might be some change in the pattern what you receive in the mass spec, as we have to try to get as much as best as possible the separation pattern in the mass spectrometer, and there are some changes from day to day a little bit, but interfacing and imaging CIEF is perfectly fine, reproducible from day to day.
Great, thanks, Christian. We have another question, which I believe is also for you, Christian. How often do you need to clean MS when coupling CIEF to MS?
Well, it's a little bit difficult to say because we are running different applications on several mass spectrometers, so we are not running a direct comparison in this way. But it looks like that there's not so much of a change if you compare to standard nano interfacing running other applications, where you always get a little bit more in than when you, instead of using a standard source where you have a stronger drying gas against the flow, and in nanospray you want to get everything in. But it turns out not to be much more. That's at least our feeling right now at the moment.
Great, thank you, Christian. So that's all we have time for today. So thank you very much to Christian, Carsten and Timon for today's informative discussion and presentation. And thank you to everyone joining us online and for your interesting questions. If you do have any other questions, please feel free to email me at editor@selectscience.net and I will follow up with your questions for our speakers. Remember, you can also download a certificate of attendance in the related resources tab to the left of your screen. And if you'd like to listen to today's webinar again or to invite someone else to listen, it will be available to watch on demand in a few days' time. So goodbye and thank you once again for everyone joining us.
Thank you.